Hello, everyone. First of all, thank you for the kind introduction and for the opportunity to be here today. My name is Rui Queiros, and I am part of the applications team at Hiperbaric. And today I'll be discussing high pressure processing, or HPP, and its impact on pet food safety, quality, and nutrition. First, I would like to mention the rest of the applications team at Hiperbaric. Currently, there are 6 of us on the team. It is spread out across 3 locations, namely Spain, US, and China. And we are a dedicated group of food scientists and provide support and consulting on HPP technology, packaging, product development, and validation studies. We are part of Hiperbaric, a company that designs, develops, and markets high pressure processing equipment, and we are proud to lead the global market in HPP technology, holding approximately 65% of the market share. With over 400 systems installed in more than 50 countries, we are supported by a dedicated team of over 150 employees and backed by more than 25 years of experience. Now, let's move on to the main topic of this presentation, HPP and pet food. HPP is a non-thermal preservation method that uses high pressure transmitted through water to achieve results similar to pasteurization, but without the use of heat. This technology ensures food safety while preserving the food's nutritional and sensory qualities. Here we can see a diagram illustrating how HPP operates. Essentially, it is a batch process, and we refer to each batch as a cycle. As you can see, a cycle begins by loading the products to be processed into carriers or baskets, and these baskets are then placed into the high pressure vessel, which is then filled with water at low pressure. Next, to increase the pressure, intensifiers pump high pressure water into the vessel, and once the predetermined pressure level is reached, the pressure is maintained for a set period of time. After this holding phase, the water is released, allowing the pressure to drop. Once depressurization is complete, the baskets and the products are unloaded and the new cycle can begin. In an HPP process, there are basically 3 main parameters that can be adjusted. One is pressure, which can be set up to 6,000 bar or 87,000 psi. Time, which doesn't have a strict upper limit, but typically ranges from a few seconds to a few minutes, usually up to 5 minutes, with 3 minutes being the most common. And water temperature, which is usually set between 5 and 25 degrees Celsius, which is about 40 or 41 to 77 degrees Fahrenheit. So when we talk about high pressure, just how much pressure are we talking about? To give you some perspective, at sea level, the pressure is about 1 bar. As you go deeper into the ocean, the weight of the water increases the pressure. And at the deepest known point in the ocean, which is around 10 kilometers deep, the pressure reaches about 1,000 bar. Now, HPP takes that even further, using pressures up to 6,000 bar, which means that's 6 times the pressure at the ocean's deepest point, equivalent to a pressure that you would experience at a depth of about 60 kilometers underwater. It's important to understand that different microorganisms have different levels of resistance to pressure. But as a general rule, the more complex the organism, the more sensitive it is. For example, parasites require relatively low pressure for inactivation. Typically, pressures a bit above 2,000 bar are enough to eliminate most of them. And yeast and molds are also quite sensitive, and they can usually be inactivated at around 3,000 to 4,000 bar. Bacteria, on the other hand, these tend to be more resistant. While some inactivation can occur at lower pressure levels, to achieve significant bacterial inactivation, it usually requires pressure of 6,000 to 6,000 bar. The viruses are generally inactivated at the same pressure levels. So the general rule is the higher the pressure, the greater the level of microbial or viral inactivation. It is also important to note that commercial HPP conditions do not inactivate bacterial spores. Because of this, additional safety controls are necessary for foods where spores might survive and grow. So we need to control them. So what are the key factors that influence the inactivation of microorganisms by pressure? First, let's look at the process itself. The main factors here are pressure and holding time, or the time under pressure. And as a general rule, and probably as expected, the higher the pressure and the longer the time under pressure, the greater the inactivation. However, beyond a certain point, applying more intensive conditions does not result in additional inactivation and still may not be productive as well. As I mentioned earlier, HPP is a batch process, so the longer the holding time, the longer the entire cycle will be, which can lead to lower productivity. Next, we have product-related factors, such as water activity and pH value. Water activity indicates the amount of free water in a product, and it is a critical parameter for HPP. For a product to be suitable for HPP, it needs to have a high water activity. For instance, products with a water activity above 0.97 work very well with HPP. However, if the water activity decreases, the efficacy of HPP also drops, resulting in lower inactivation. Below a water activity of 0.87, there's almost no inactivation. In summary, products with very low water activity are not really suitable for HPP. Another important parameter is the pH. Basically, pH and HPP work in synergy, meaning that the lower the pH of the product, the higher the bacterial inactivation achieved by pressure will be. And lastly, as I mentioned earlier, different microorganisms, including various species and strains, have different levels of resistance to pressure, and that needs to be taken into account. So what are the applications of HPP, or in other words, which type of foods can benefit from this technology? HPP is a versatile technology that can be applied to a wide range of foods, including juices and other beverages, various meat products, avocado-based products like guacamole, other types of salsas and dips, as well as baby food. Other food categories that work well with HPP include ready-to-eat meals, seafood, dairy products, and of course, pet food. In all of these categories, HPP helps improve food safety and extend the shelf life without compromising quality. Before finishing this introduction to HPP, let me just share an overview of the HPP market, so you can get an idea of the main applications, or at least the percentage of machines processing for each category. The primary product being processed with HPP with nearly 24-25% of machines in operation is dedicated to juice. Juices are followed by meat products and guacamole, each accounting for nearly 20% or less. And it's also worth noting that a significant percentage of the machines are installed in tolling companies that are businesses that may not produce their own products, but offer HPP services to their clients. The remaining machines are distributed across the other applications that I mentioned earlier. Now, moving on to the main topic of this webinar, let's take a closer look at HPP pet food applications. Just a quick note, for this presentation, I've gathered research studies from third-party researchers. So please note that the research that I'm sharing is not our own. All references used are cited in the respective slides in case you want to check them for further information. To provide some context, what are the risks associated with raw pet food? Well, raw pet food diets carry risks due to the harmful bacteria such as Salmonella, Listeria, and E. coli. These pathogens can contaminate the food and spread to both pets and humans, which can lead to serious health concerns. Several product recalls highlight the ongoing safety issues with raw diets, highlighting the need for stronger safety measures. So, why can HPP be the solution? Well, HPP is an FDA-accepted method that achieves a 5-log reduction of pathogenic bacteria, meeting the requirements of the Food Safety Modernization Act. And this process not only improves safety and extends shelf life, but also preserves the raw food benefits that pet owners value. The first study I would like to share evaluated the effectiveness of HPP on a chicken-based pet food formulation, which contains more or less 65% chicken meat, about 20% chicken organs, and the rest is made up of fruits, vegetables, and other ingredients. And in this study, HPP was applied for 2, 3, and 4 minutes at nearly 6,000 bar, or about 85,000 psi. The researchers inoculated the formulation with cocktails of pathogenic strains of Salmonella, Listeria, and E. coli, and reached an initial bacterial load of around 7 to 8 log. As I mentioned, they evaluated different holding times and monitored the changes during frozen storage. The results show that HPP is highly effective in pathogen control, achieving reductions of over 7 logs for Salmonella and E. coli under all the tested conditions, meaning that applying pressures at this level for as little as 2 minutes at low temperatures will guarantee the safety from these particular pathogens. However, the initial inactivation of Listeria was just below a 5-log reduction. Despite this, the treatment was still enough to cause sublingual damage to the bacteria, allowing them to eventually die off a couple of days later, as you can see in the graphic. In this particular case, a 3-day hold would be necessary before the product could be safely commercialized. It is important to note that, in general, increasing the holding time from 2 to 4 minutes improved Listeria inactivation at all sampling points and days. While HPP typically achieves 5-log reductions for all the 3 pathogens after processing, I believe it's important to showcase these examples to emphasize the need for validating both product and the process. Nevertheless, HPP constantly delivers superior pathogen inactivation compared to other non-thermal methods, ensuring higher food safety. Here, another study by the same authors showed that the specific formulation of pet food also influences how pathogens behave during processing. In this particular study, different ingredient mixes were tested, labeled as formulas A, B, and C, each with slightly different pH, water activity, and nutritional profiles. And these variations provided a good insight into how HPP interacts with different types of ingredients. The most notable difference among these formulations is the protein content. Formula A has the lowest protein percentage, about 14 or 15%. Formula B at 15%, and Formula C, which contains the highest protein level, around 70%. This chart highlights how all three formulations responded to HPP treatment over the course of frozen storage. Basically, the combination of pressure and low storage temperatures significantly reduce the three pathogens, confirming that HPP remains effective across different recipe types and compositions, especially for Salmonella and E. coli. In the case of Listeria, we can see that higher protein content seemed to protect the bacteria, with lower inactivation observed in Formula C, the one with the highest protein, followed by B and then A, which achieved with this one that achieved the 5-log reduction. Although not shown here, higher fat content may also offer some protection to bacteria against pressure. All of this reinforces the importance of formulation in HPP products in general, and particularly in pet food. One way to further increase pathogen inactivation is by adding, for instance, lactic acid fermentate to raw pet food and combining it with HPP. In this particular case, only 0.7% of lactic acid fermentate combined with nearly 6,000 bar of pressure showed an even greater effect, particularly for Listeria. In fact, it achieved over a 5-log reduction, if I'm not mistaken, in just 2 minutes of HPP treatment. This shows how lactic acid and high pressure can work together to further enhance food safety for raw pet diets. As a quick side note, I just want to mention how HPP affects proteins. High pressure primarily alters the tertiary and quaternary structure of proteins. There is the three-dimensional folding and the arrangement of subunits. It doesn't break the actual peptide bonds, so the nutritional value of the proteins remains largely intact. Still, at higher pressure, irreversible changes can occur, but these do not compromise protein digestibility or nutrition for pets. However, it is important to note that pressure does affect these higher level structures, as we will explore shortly. Beyond bacteria, HPP is also effective against viruses. High pressure can disrupt virus envelope, denature viral proteins, and even destabilize its genetic material. These effects destroy the virus' ability to infect cells, which is especially valuable when addressing concerns like avian influenza contamination in pet food. And it is important to highlight that many key structures in virus are protein-based. And as we saw in the previous slide, pressure alters the higher structure of protein. Therefore, it can change the structure and function of these viral components, damaging the structures that viruses use to attach to host cells and rendering them inactive. Sadly, there aren't any studies specifically about the effects of HPP on the avian influenza virus, particularly the H5N1 subtype on pet food. However, there are a couple of studies on the H7N7 subtype, which is quite similar to the H5N1 and may provide us with some valuable insights. For example, when cell culture medium contaminated with this virus was processed at 4,000 to 6,000 bar, the viral count dropped dramatically, with more than 5 log reduction occurring quite rapidly. In fact, in chicken meat, 5 log reduction was achieved in under 30 seconds at 5,000 bar, which demonstrates that HPP can neutralize viral threats very quickly and efficiently. Just like to mention that another study reported similar results, namely 5 log inactivation at 6,000 bar for 60 seconds, also in chicken meat. It is important to note that HPP typically uses these high pressure levels, that is 6,000 bar, for longer than 60 seconds, which should be more than enough to control the virus. The authors of this study also identified the pressure, time, and temperature conditions required to achieve 5 log reductions of the virus. So these graphs illustrates how pressure and temperature work together to inactivate in this case, the H7N7. The blue rectangles that you can see on the left on the graphs highlight the pressure and temperature range typically used in commercial HPP machines. And as you can see in both cases, at 5,000 bar, it takes only 50 seconds, the blue line, 15 seconds under pressure to achieve a 5 log reduction, both in cell culture medium and in chicken meat. The authors also demonstrated that even at around 4,000 bar, a 5 log reduction is still achievable, provided that the holding time is extended to 2 minutes. These findings reinforce the value of HPP as a versatile and effective tool for virus control in pet food. However, despite these promising results, validation using the H5N1 subtype is still necessary to fully understand the HPP effectiveness against it. Well, what about nutrition? Several studies have already evaluated the impact of HPP on the nutritional value of different foods. This particular study looked at the effects of HPP on the macronutrient profile of 3 different pet food formulations. You can see the ingredients on the top of the graphs, each with varying levels of protein, fat, carbohydrates, and other components, and found no significant differences when compared to untreated raw foods. While these graphs show some slight variations, these differences were not considered statistically significant, which means that pet owners can feel confident that the nutritional value remains stable even after HPP treatment. Regarding micronutrients, a pet food manufacturer that uses HPP analyzed the effects of pressure on the micronutrient profile after processing and published the results. The study found that micronutrients, including amino acids, fatty acids, and minerals, remained largely unchanged, with variations of less than 0.5% after HPP treatment. Only for vitamins, namely vitamin A, B5, B6, and B12, show some measurable losses. However, these reductions were not significant enough to fall below AFCO's recommended nutritional levels, as these vitamins remained their concentration at least 10% above the required minimum. Nonetheless, to ensure maximum nutritional value, the company chose to restore the original vitamin levels by simply adjusting the ingredient proportions. In this particular case, they increased the inclusion of raw liver, organ meat, and muscle meat. And like I said before, adjusting formulations is maybe necessary and is an important part. Another important parameter that has been studied is whether HPP affects the digestibility of food or pet food. This chart shows the results of a study comparing the digestibility of chicken meat after different processing methods, both heat and pressure. When looking specifically at the protein digestibility, HPP treated food maintained almost the same digestibility as untreated raw food, with the small differences that can be seen here not being significant. In contrast, traditional heat treatments, such as cooking at temperatures between 70 and 121 Celsius, significantly reduce the digestibility. This highlights that HPP preserves bioavailability of nutrients much better than conventional thermal methods. Let me just change here, okay. In live animal trials with 10 clinically healthy male dogs that were fed for 45 days with HPP pet food, the main results showed that these dogs had much higher nutrient digestibility, between 22 to 26% of digestibility, compared to dry food that only reached about 57%, I think. Dogs also produce less feces, which is another strong indicator of better nutrient absorption. So we can say that HPP isn't just safe, it supports healthy digestion too. One of the key advantages of HPP is its minimal impact on the look and texture of food. A recent study demonstrated that even at high pressures, changes in the color and appearance of a chicken-based pet food formulation, which contains around 12% protein, these changes were minimal. As you can see in these photos, the product treated at two different pressure levels looks nearly identical to the untreated version. So on each image on the left, you can see the no HPP version, and the HPP version on the right, you can see the differences are very small. Which means that pet owners receive a product that looks fresh and appealing, while pets enjoy the taste and texture they expect. HPP can be applied to a variety of pet food formats, although one of the most common approach is to process the final formulation in its final packaging, which is then sold either frozen or refrigerated. And another common option is to further process the HPP treated product into a clean room, shaping it into bite-sized pieces that can then be freeze-dried and sold shelf-stable. As you can see, there are several ways to leverage the benefits of HPP in pet food processing, depending on the desired product format and the shelf life that needs to be achieved. Here are a few examples of companies that already are using HPP to enhance safety of their pet food products. I'm sure you recognize most of them. And I would like to finish by mentioning that you can find our pilot plants at these three locations, namely Spain, United States, and China, which are available for complementary HPP testing, or for any doubts that you may have, you can contact us. Feel free to just visit. And then the quick mention to the Pet Food Forum. There you will have a great opportunity to learn more about this topic through a couple of talks that are on the screen, or to schedule a meeting with our team. Just follow the QR code also on the screen. Sorry. And that's all I have to say. I hope it was helpful, and thank you for the attention.
Thank you so much. And before we do the Q&A at the end, we have a second speaker. So we're going to go through her presentation, and then the Q&A will be together at the end, just for audience information. So our second speaker today is Dr. Grace Denow. Dr. Denow is a research associate professor in the Department of Food Science and Technology at the University of Nebraska-Lincoln. Her research interests are in the broad area of value-added processing, focusing on HPP, high-pressure homogenization, and freeze-drying. Since 2017, she's coordinated the HPP and Engineering Services Laboratory of the Food Processing Center at UNL. She and her team conduct microbial challenge, process optimization, process validation, and shelf life studies that have helped the U.S., and more specifically Nebraska, food manufacturers comply with USDA, FSIS, and FDA food safety regulations and bring their products to market. At UNL, Dr. Denow teaches a class in pet food manufacturing to support the new undergraduate minor program in food technology for companion animals at UNL. All right, Grace, the presentation is all yours.
Well, thank you so much, Lindsay, and thank you so much to Hiperbaric and Dr. Queiros for giving me the opportunity to speak a little bit about a really fun project that I've decided to undertake with some partners, both from industry and academia, to kind of just advance HPP research. So the consortium right now, when you kind of think about it, is kind of naming it as the High Pressure Processing Industry and University Cooperative Research and Resources Program. So from Lindsay's introduction, most of you know that my lab does do quite a bit of testing, not just necessarily on pet foods, but any kind of food materials, both humans and animal foods, that utilizes HPP, whether or not it's for pasteurization or to kind of maybe leverage other cool effects that you might get out of high pressure processing. Certainly my lab also does quite a bit of training when it comes to disseminating some of our own research or research that we might find in the literature, or even just like, the basic principles of HPP, because I definitely think that, the more people who learn about this technology provides them with another option they can use to processing their product and perhaps get to the level of safety or to the level of quality that they're trying to achieve. So with this consortium, if you will, we're right now still in the process of forming the consortium and really looking for some industry partners. As an academic, I can probably always look through the literature and figure out where some of the knowledge gaps are when it comes to a particular topic. But I think with HPP or similar food processing technologies that are up and coming, I think it's really important to keep in mind end users. So in this particular case, it will be food industry partners, and kind of figure out, like, how do they want to use HPP and what are some of the questions that they want answered, whether or not if it's on the safety side or on the quality side. And then that's where we would tailor our own research topics. So for this particular consortium, the very first project that we decided to kind of tackle together is the whole issue of avian influenza, or specifically H5N1, being a new non-foodborne hazard that we're going to have to control for in pet food manufacturing. So as I think some of you who've talked to me or called my office in the last couple of months, very first thing when we started to look at or find out that there were several cats that were either getting sick or dying because of exposure to H5N1, and more importantly, looking at exposure potentially through food that they have consumed, the very first thing that people came to me was like, Grace, we need validation studies. And my initial reaction was, oh my goodness, this is going to be really hard, because when you're working with H5N1, something that's highly pathogenic like this, and even a select agent, you're going to need to have a BL3 laboratory or biosafety level 3 lab. In the United States, we only have one lab that I can think of that has both BSL3 and access to an HPP equipment. Most of us, or the rest of us who have access to HPP equipment, are only utilizing or only have access to a BSL2 lab. And it also goes with most of our commercial food testing laboratories. So I kind of hesitated really kind of doing some validation studies in this particular area, because first things first, I kind of need to understand the space, like what exactly is going on, what are the challenges, what are perhaps, the direction regulations are going to go into. And so that's eventually when I realized, again, as an academic researcher, I can't really solve this on my own. I really need to pull in industry here, and then they can dictate a little bit more about what they need first before they even get to designing a validation study, and also where is regulatory kind of moving towards, because we at the end of the day we want to make sure that we're also going to meet regulatory requirements, and also probably help then advance knowledge in this particular area and fill in some gaps where we didn't know that we had to know before. So I know Rui had mentioned earlier, unfortunately, there's just has not been that many studies with HPP and avian influenza in the past, and even then, only H7N7 subtype has been tested. We're glad to have those studies, but again, this is a fairly new hazard. It's also not something that's foodborne that we would normally think of, but we should recognize from the literature, it's well established that cats, both big and small, are really sensitive to this particular virus. So first thing that came to my mind was, okay, well, we need to kind of understand which are your most risky ingredients. So looking at your supply, and if you look at the FDA guidance or document that says pet food companies need to reanalyze their food safety plans, include H5N1 as a new hazard, then one of the things that they said was, you can certainly look to coming up with ways through supplier control to perhaps mitigate this particular risk or hazard in your manufacturing. So some of the salient points that I've learned through engagement with some of you from industry have been, well, yeah, definitely a lot of folks went back to their suppliers and started to just kind of requiring some letters, trying to understand, when were certain flocks of birds perhaps were tested for H5N1, what was the elapsed time from that date of testing to the date they were actually processed, to the point of me receiving this particular lot of ingredients. So I think that's really a good starting point. One of the things that the consortium is working towards is perhaps coming up with additional guidance on, if you're going to rely solely or mostly on supplier control, what are some of the other questions that you should ask your supplier to provide, so that you can have a little bit more reassurance as well that the ingredients that you're getting are going to be free, or at least have a very reduced viral load. The second thing that, again, after discussing with colleagues from industry, was we realized that there really needs to be a better risk assessment out there when it comes to raw, specifically for raw pet food manufacturing, those that are not using heat at all to control for the virus. So in this particular case, in that risk assessment, we want to understand which ingredients tend to be more risky than others, and is there anything that you could do perhaps as a pre-treatment of those ingredients prior to receiving them into your facility. I think this is like really unknown territory when we're starting to look at what could potentially be the viral load. Because when you think about it, perhaps even in a flock of birds, maybe by the time that they're getting tested, maybe only 1 or 2 birds might be sick at that point, and then they get cleared to being processed, because we can't necessarily test every single bird. So all of a sudden there's a little bit of a dilution effect to try to find this needle in the haystack, so to speak, to find the very few viruses that might be in a particular flock. So, and then with everything else that you can do, whether or not it's through sanitation or doing perhaps some acid treatment, maybe doing a blanch treatment on some of your ingredients, then all of that, every little thing that you do, you might want to consider them as, like, let's say a hurdle technology, and then still have some kind of a kill step in your process. So we also kind of want to understand, like, when you're trying to do some of these hurdles, where do you get the most benefit of doing them? Is it going to be before your kill step or after your kill step? So in this particular case, there's definitely going to be some kind of a predictive microbial or viral reduction model that we're going to have to do, hopefully as a web-based tool that could really help manufacturers figure out how they're going to revise perhaps their processing in order to control for this particular virus. I did put a picture here of a dairy cow being milked, because I think compared to chickens, dairy cows are really, through their mammary glands and through the milk supply, are really shedding the virus at much greater numbers than, let's say, in a single chicken or a single turkey bird. So there, I think we can definitely rely on heat pasteurization as a means to kill such a high, heavy load of the virus. But that's not necessarily, I know some pet food companies don't necessarily want to do a heat pasteurization step. So there still needs to be some evaluation of HPP for that particular application. And I think that's something that the consortium would like to look at in the future. Once we actually get through discussing, how you're going to do a supplier control, the next set of questions really are, well, how are we even going to test for this particular virus? Perhaps not just in the finished pet food product, but then also with the ingredients in your supply chain. So the hardest part about testing right now is that question, do you test your ingredients or do you test the finished product? My suggestion at this point really is understanding that the current PCR tests are being used by veterinary diagnostic centers to detect a sick animal. You're going to get those tests are really designed to look at single proteins. So that means really looking at your ingredients more so than a complete and balanced diet, perhaps, or a complicated food matrix. Because what people are also finding out with these particular test kits is, number one, there could be an issue with how do you extract such a small amount of virus from a particular, let's say if it's been like a formulated diet, then there's definitely a dilution effect happening. Any other kind of ingredients that you throw into the mix might also cause an interference. So I'm going to call that the matrix effect. So this particular virus is, all influenza viruses tend to be wimpy. They're pretty easy to kill, but we've never really thought about killing them in foods. But we can do so with heat. So testing is going to be a big thing. So then I started to think about, it's kind of like the chicken and the egg problem, which has to come first? Do we have to solve the problem with testing first, or should we go ahead and solve the problem of the lack of validation studies? I think for the most part right now, because there's also some testing issues with validation studies, we definitely need to also look, start looking at and working on the testing issue. So a couple of tests do come in mind that are pretty used when looking for the virus, and there are three that I've highlighted here. The first one being PCR. So talking to my colleagues in the Veterinary Diagnostic Center and then also a virologist, they told me essentially like PCR is going to be your most sensitive test. That's going to give you some quantification, especially if you use a quantitative PCR technique. You can get some correlation to the actual viral load in there. But as most of you know, this is still a presumptive test. We don't know necessarily whether or not the virus that's in there and in the food material, the food itself is not a suitable host. So the virus is not actively replicating, but it's there. And if it can cause harm to an animal or human, it would have to be present in high numbers. But for this particular test, though, if you test positive in a PCR test, there's still a little bit of an unknown whether or not is that virus going to be active, meaning that when it is consumed by a suitable host, will it cause some harm? So I know the FDA is going through a different route, but there's still a part of me that thinks, well, maybe there is a need for some kind of a confirmatory test. Maybe not for normal monitoring of the process or when what's being released to the marketplace, but we need some kind of a confirmatory test when we get to a validation study, because in a validation study, kind of similar to the two papers that Rui highlighted in his presentation, we're going to need to know after HPP, was that live virus that we inoculated in the food product also remain active, even though perhaps in a reduced load or reduced numbers in the finished product or ingredient, is that still going to be able to cause harm? So that's where this picture where you see some Petri plates in there. This is where some plaque assays could really become very, very helpful. So in a plaque assay, you have a cell culture. So an appropriate cell culture might be bovine mammary cells, because we know that dairy cows in their mammary glands that's really a harbor site for the influenza virus. Or perhaps, I'm not yet sure, but perhaps some kind of a feline intestinal epithelial cells could be the appropriate cell culture. Then when you do extract a virus from a food material or an ingredient, once you inoculate that cell culture with this, if you have some active virus that are still there, it's going to kill those cells, and they're going to present itself as little circular plaques. So then you can count the number of plaques that are formed. The higher the number of plaque forming units, you can kind of think of them as colony forming units if you're more familiar with bacterial testing. Then that could also give you an indication of whether or not you have a high viral load that is also pretty active, that could definitely infect bovine mammary cell cultures or even feline intestinal epithelial cells. So a lot of this, I think, is still somewhat on a research stage or phase, not necessarily fully validated yet. There's no ISO method that we can kind of push reference to. But I think this is one really cool thing that we can work with pet food manufacturers, maybe some of your commercial testing food laboratories, and also university to kind of solve this particular problem. And it's something that is very high, I think, on the priority list for our consortium. And then finally, the last one I put in there is an egg, and we can kind of think of it as a chicken egg that has an embryo in it. This is actually the gold standard when people want to try to isolate a particular virus. You grow the virus, let it replicate, then you inoculate it into the egg, and if it happens to be live and can cause harm, then within a matter of days, then it can kill the egg. Both the plaque forming units test and the embryonated egg test are not as sensitive as PCR. The reason for that is there could be other things in the food matrix that actually kills the virus first before it even makes contact with, let's say, the cell culture or the embryonated egg. So I think a combination of these three things is actually what we're going to have to evaluate and really allow us to better understand how we're going to monitor or test for H5N1. And then, but certainly the picture I had earlier with the centrifuge tube there, we're also going to have the challenges or face challenges when it comes to extracting the virus in the first place from any kind of food material. And then finally, you know, not necessarily sequential, but while we're doing all of these kind of attacking or addressing needs for looking at how to do a better supply control, doing a better job of extracting, do a better job of testing or evaluating those different kinds of tests, then I think we should have an easy win or a first win when it comes to doing an HPP validation study. So perhaps, you know, from our consortium at least, we're thinking of doing some simpler validation studies to begin with, not necessarily using a complicated, complete and balanced diet food matrix to begin with, where there are so many different ingredients that could potentially interfere with our testing and on the efficacy of HPP, but maybe starting off with a very simple ingredients. So let's say chicken livers, which are ubiquitously used when it comes to, like, let's say a pet treat or as an ingredient in a lot of pet food diets. So that's essentially what I wanted to talk about today, or at least give everybody a chance to see perhaps from university what are some of the activities that we're doing in order to help the industry, help regulators to kind of fill in some gaps in the literature and really advance not just our knowledge on testing for H5N1, for pet food safety, but then, you know, down the road, this could also have implications on the human food safety. And also, I think it's just kind of like a really neat project for everybody to be more transparent about how they're using HPP to make sure their product is safe, but then also what are the other ways that you can do to make raw pet food safe. So with that, I'm going to go ahead and turn it over to Lindsay and Hui, and I think we have some time for questions.
We do indeed. So let's get both our speakers back on camera. And I have questions going all over the place, so we will see what we can do. So the first question I have is HPP effective for freeze-dried products?
So I can start that off. I do know that a lot of raw pet food manufacturers will HPP first, because that is a kill step that, you know, you can have a validation study that demonstrate a 5 log reduction in the 3 pathogens that Hui mentioned earlier. The reason for that is freeze drying is not necessarily a kill step. It usually is pretty good at preserving bacteria and viruses. Depending on how you program your freeze dryer, you may be able to get a 1 to 2 log reduction, but that's still definitely far from the requisite 5 log reduction. So yes, HPP is effective when it comes to freeze dried raw products.
Okay. And then considering that H and N proteins from avian influenza virus only play a role in the attachment of the virus to the host cell and in the release of the virus from infected cells, would it be correct to assume that H7N7 and H5N1 subtypes would behave similarly against HPP? They're asking if the pressure resistance is comparable.
Well, I would say yes, but we do need the research for confirming it. But yes, I believe so. Like I said, HPP affects the structure of proteins, so you would change the conformation of the attachment points. So in theory, it should work, but well, we need the testing.
Yeah, I would also agree in general. Like, I think the H and N kind of portions of the virus really just kind of encode for how the virus is going to replicate, what kind of proteins are going to be made. But overall, you know, viruses typically are easier to inactivate with compared to bacteria in HPP. And the reason for that is just a much simpler structure because they're mostly made of proteins. And as Dr. Queiros already mentioned, HPP is really good at changing the conformation of these proteins up to a point that they don't work.
That makes sense. This question is for both of you, since you both talked about it from your respective perspectives. You talked about the survival scenarios of pathogens in HPP, like higher fat content, more protein, and that there could be some pathogens left over. And then Grace, you talked about a combination of hurdle and kill steps to like really ensure everything is getting sorted. Have either of you found that there's like a magic combination of hurdle and kill steps involving HPP that exists, or is it all very case by case? Like, let's see what's left after HPP and go from there.
Yeah, I would say it really is case by case, because I mean, I can even speak to one particular case scenario that we had to deal with during the pandemic. So somebody was using a combination of phage and then also HPP in order to get to their 5 log reduction of salmonella in this particular case. And then during the pandemic, when it was really hard to source from their main supplier their poultry ingredients to begin with, they switched suppliers. And so by switching suppliers, it actually ended up coming in with a much higher salmonella load to begin with. So then what they were doing as a combination of phage and HPP was no longer working for them, because even after what they used to do and then being able to test negatively for salmonella afterwards, this time around they were getting a lot of positive hits. So in that particular case, yes, you can have kind of like the same combination of, let's say, a hurdle plus HPP or kill step, but then all of a sudden when you change a supplier, not even a change of ingredients or formulation, it doesn't work anymore, right? So that type of thing, kind of just demonstrates that it is really case by case.
Yeah, I agree. And even if you consider that sometimes you have the ingredients that can have an antimicrobial effect that will play into the inactivation as well, then you can have different processing conditions by 1 minute or two that can inactivate even more what you would get without that extra minute. It should really be a case-by-case study, just make sure, because overall we know that HPP lets you get those high inactivations, but there are particular cases where the things need to be adjusted.
And can HPP be used twice? Like, can you run something through HPP and then do it again? Or is it like a one and done, it won't do any good to do it again kind of thing?
Yeah, you can do it twice.
Yes, and I think there's been some cases before where, when you think about it, the first time you HPP a particular food product, it's going to sublethally injure those cells. And as Rui mentioned, if there's anything in your formulation that can continue to stress those cells, or let's say you're freezing after HPP, changing immediately the temperature afterwards, right? The formation of ice can further stress those sublethally injured cells to the point that they die. So in some of the slides that he showed, you can kind of see that over time, throughout the length of storage, frozen storage, you can get a little bit more killed each day, because that's kind of what's happening. Now, if you imagine you can HPP a food product that's ever been HPP before, you sublethally injure those cells. So you've kind of punched them once, and then you put it through the HPP immediately afterwards, and you punch them again, then yes, you're definitely going to get a lot more kill this time around. But then it's also the cost of time and money to have to do it twice. So those are some of the maybe economic balances that you're going to have to do.
Yeah, basically the compression and decompression phase exerts more stress to the cells than the holding time. So doing it twice quickly, you have twice the effect of the compression and decompression. But yeah, that's right. That takes longer. That damage more the
Equipment.
The equipment, and overall it makes the process longer and more expensive.
Yeah, that makes sense.
But it works really well.
I have a question about PCR testing. Can PCR give false positives for HPAI? What if the virus isn't active, but PCR still detects the presence of genetic material?
Yes, and that's really what's happening, right? Is that you are really testing for whether or not you can find that viral RNA in there. So these days, like most of the time, at least in the veterinary diagnostic world, they're testing to see, especially if they're seeing symptoms of the illness, this respiratory distress. So there's already a clue to them that, okay, this might be H5N1 or avian influenza. So then they would test with PCR to see if they can find the viral DNA or RNA in there. And then that would kind of confirm for them whether or not there's this is an H5N1 case. In our particular case in food testing, right, like if we're just pulling food from the shelves and then testing them, we don't necessarily have an indication yet that somebody has gotten sick by consuming this particular food. So for us, by finding that particular viral RNA, we don't necessarily know if it's active or inactive. So that's going to be what we would then consider presumptive. So we will have to then figure out if this were bacteria, to do a confirmation test to see if it'll grow on some kind of a media, because then that would tell us that, oh wow, this bacteria is actively growing. But then for the virus, it makes it really tricky because we need to figure out if there is something comparable to that, but there are challenges because the virus is very easy to kill. It's very, I call it wimpy and such. And then it's so small in numbers, right? Finding that needle in the haystack. So this is why I think regulation right now for the FDA, they've kind of gone through a different path where they're doing a whole genome sequencing to see if they can identify that very same virus in a cat that is sick, or a cat who has expired. So yeah, there's still some challenges that we have to look into, but you're absolutely right. It detects the viral RNA, but does not necessarily tell you immediately whether or not it's an active virus that could cause harm.
And Rui, you mentioned the standard volume of runs with HPP. Is there a means of being able to use a large volume or a continuous process to be able to do larger runs with HPP?
Well, the best way to process larger volumes is to adapt your packaging to the shape of the high pressure vessel. So if you optimize the shape of the packaging, you will increase the vessel filling ratio, and with that you can process more than you would if you had a shape of packaging that wouldn't fit perfectly into the vessel. For liquids, we have also a bulk machine that processes liquid food, and it's still a batch process, but I would say it's a semi-continuous one with a filling ratio of about 90%, and that's allow for faster and in higher quantities amounts of product. But yeah, there are ways to improve the capacity of production of the equipment.
Great. And we are about 7 past the hour, so I have one final question for the both of you. From the perspective of as regards avian influenza and then just kind of overall in the pet industry, what do you think is next for the technology of HPP? Is there anything untapped, anything currently being researched? What's coming down the line with regards to avian influenza and then overall?
Okay,
Go ahead first.
Okay, yeah, I can get started. Yeah, I think overall for avian influenza, it's not just my group, but there are other groups that have access to both, even if it's low pathogenic avian influenza, so one that doesn't require a BSL-3 lab, but those that also have access to HPP, there's going to be a couple more research studies that involve validation. And I think those are really great. I think the more of us that can kind of do that and add to the body of literature, the better. When it comes to just foods in general or and stuff, I think there's now like this movement, or at least a desire to see what else can we do with HPP, leverage high pressure processing. Even if it's not going all the way to the 6,000 bars or 87,000 PSI for pasteurization. So there's a couple of us researchers that are looking at, well, we know starting at 300 megapascal or 3,000 bars, and maybe even all the way up to 500 megapascal or 5,000 bars, we know that we can start modifying the structure of proteins and perhaps modifying their functionality. So is there something there then that we could leverage towards improvement of, let's say, quality of a food material? So I think you're going to see a couple more applications that go for quality rather than just food safety.
Well, from our side, not particularly in pet food, but in food in general, what we are seeing is an interest, or a growing interest in combining high pressure with high temperature, so high pressure thermal processing. This is not non-thermal anymore. but it allows for better products that are exposed less time to temperature, which means that you can get a shelf-stable product without the thermal load being as high as a traditional thermal process. I think it's the thing that is getting more inquiries at the moment.
Yeah. Yeah, I think that would be great to be able to control for, let's say, Clostridium botulinum and its spores by a combination of high pressure and then a very short high thermal treatment along to go with that high pressure would again kind of allow these HPP treated products to enter a different part of the grocery store.
Yeah.
Well, thank you both so much. If anybody in the audience has further questions, their email addresses are currently on the screen. Feel free to reach out to them. I'm sure they would love to speak with you after our presentation today. I would like to thank you all for taking the time to listen to today's webinar. Remember that an on-demand video of this presentation will be made available to view starting tomorrow. To register for some of our upcoming webinars, view the on-demand video of today's webinar, or see on-demand webinars on related topics, visit petfoodindustry.com/webinars. Please remember to take a couple of minutes to answer our short survey after the webinar and let us know what you think. Thank you to our sponsor, Hiperbaric, for their participation today. For more information on their products and services, please visit hiperbaric.com. Finally, I'd like to extend a big thanks to both of our speakers today. Thank you again for joining us, and have a great day.
Thank you.
Thank you for having us.